students, we are discussing plant breeding, right? In plant breeding chapter, we started introduction, right? Now, what is the meaning of plant breeding? It is a purposeful manipulation of a plant. Purposeful manipulation of a plant. It means with a purpose we manipulated a plant. By which what is produced, sir? A desired plant species is produced. This plant species, it is suitable for cultivation. It is a high yielding. And what more, sir? Disease resistant. What? Disease resistant. Is it okay? Right. Now listen here. Conventional plant breeding. What it is? A conventional plant breeding. We can call this conventional plant breeding even as a classical plant breeding. Conventional plant breeding or classical plant breeding, right, has been carried out from thousands of years since start of human civilization. Is this statement okay? Conventional plant breeding or classical plant breeding has been carried out from thousands of years since start of what, sir? Human civilization. But according to recorded evidences, there is a recorded information. According to what, sir? According to, right, recorded information. According to recorded information, right, it dated back 9,000 to 11,000 years. Is this statement okay? Therefore, according to what? Recorded evidences, recorded information, right, it means conventional plant breeding dated back, right, 9,000 to 11,000 years. 9,000 to 11,000 years. Is it okay? Right. Now, listen here. We are discussing, right, plant breeding, right? Now, listen here. Sir, today, what crops we are using, right? Most of the crops of today, they are a result of domestication that was done in ancient times. Most of the what? Crops. Most of the crops of present days are a result of domestication. In ancient times. Sir, what does this statement mean, sir? In ancient times, what was done? Domestication was done. Due to the result of the domestication only, right, we got many crops what we are using in present days. Is this statement okay? Right. Now, sir, what is the meaning of domestication? What is the meaning of domestication? Initially, all plants are wild plants, yes or no? It means without involvement of human beings, That the growth of that plants has taken place. It means such plants are, which plants are? Wild plants. Without involvement of human beings, if growth of a plant takes place, we can call that plants as wild plants. Slowly, human beings, what they did means, they selected some plants which are useful to them, and they started cultivating such plants. Initially, all plants are wild plants. In that wild plants, human beings selected some plants which are useful to them and started cultivating such plants. It means now wild plants are brought under what, sir? Human cultivation. It means now these plants becomes, which plants are? Cultivated plants. Therefore, write bringing wild plants under human cultivation. Bringing what? Wild plants under human cultivation is called as domestication. Is this statement okay? Right. Therefore, what is the meaning of domestication? Bringing wild plants under human cultivation is called as domestication. Right. Take animals. Initially, animals are also wild, right? Therefore, wild animals. Human beings, they started rearing some animals. Yes or no? Therefore, bringing or rearing, right, wild animals is also called as what, sir? Domestication. Therefore, right, human beings selected some wild animals and they started rearing that wild animals. Yes or no? Therefore, now this process is also called as what? Domestication. Is it okay? Right. Concerned to plants, bringing wild plants under human cultivation is called as what, sir? Domestication. Is called as what? Domestication. Right. Now listen this statement. Today, all our major food crops, are derived from domesticated varieties. Derived from what, sir? Domesticated varieties. Is this statement okay? Right. Therefore, today, right, what food crops we are using, all our major food crops, from where they are derived from? They are derived from domesticated varieties. They are derived from where, sir? Domesticated varieties. Derived from domesticated varieties. Is this statement okay? Right. Classical plant breeding involves classical plant breeding involves right, number one, hybridization. What it involves, sir? Hybridization. What it involves? Hybridization. Followed by artificial selection. Followed by what, sir? Artificial selection. Followed by artificial selection. Sir, classical plant breeding. What is another name? Conventional plant breeding. What it involves? Number one, hybridization. What it involves? Hybridization. Right. After hybridization, what we are doing, sir? We are doing artificial selection. First hybridization followed by artificial selection. Is this statement okay? Clear? Right. Now listen here. Sir, we are discussing plant breeding, right? With advancement in with advancement in genetics, molecular biology and tissue culture, at present we are using plant breeding with molecular genetic tools. With what? Advancement in development. With advancement in what? Genetics, molecular biology and tissue culture, with advancement in what? With advancement in genetics, molecular biology and tissue culture, right? Plant breeding. Plant breeding is carried out. Plant breeding is carried out by using molecular genetic tools. By using what, sir? Molecular genetic tools. By using molecular genetic tools. Is this statement okay? Right? Now listen here, sir. If we are a plant breeders, then what are the characters we will expect in a plant, sir? What are the characters we are going to expect in a plant? What are the characters we expect in a plant? A plant breeder, right, expect following characters. In a plant. Sir, what are the characters a plant breeder is expecting in a plant, sir? Right, number 1, go for the first character. Increased crop yield. What? Increased crop yield. Number 2, improved quality. Improved what, sir? Quality. Improved quality. Number 3, tolerance. What, sir? Tolerance to environmental stress. Tolerance to what, sir? Environmental stress. Sir, environmental stress, what it indicates, sir? Environmental stress such as salinity, extreme what, sir? Temperatures. Extreme what, sir? Temperatures. Extreme temperatures. Is it okay? Next, number 4, resistance to pathogens. Resistance to what, sir? Pathogens. Resistant to pathogens. Sir, pathogens means which causes diseases, right? Pathogens means which causes diseases. Pathogens such as bacteria. Pathogens such as bacteria, virus and fungi. Is it okay? Right? Therefore, see these statements. If we are a plant breeder, what are the characters we are expecting in a plant means number 1, increased crop yield. Number 2, improved quality. First one is concerned to quantity. Second one is concerned to quality. What is the third one, sir? Tolerance to environmental stress. Tolerance to environmental stress such as salinity, right? And what more, sir? Extreme temperatures. Okay, right. Next one is a resistance to what, sir? Pathogens. Resistance to what, sir? Pathogens. Resistance to pathogens. These are some characters what we expect in a plant if we are what, sir? Plant breeders. If we are plant breeders. Now listen this statement. Even we can expect another character. What it is? Tolerance to insect pest. What it is? Tolerance to insect pest. Tolerance to insect pest. Tolerance to what, sir? Insect pest. Is this statements okay? Right? Now listen here. Sir, we are discussing plant breeding. Yes or no? This plant breeding program is carried out in a systematic way throughout the world. Carried out in a systematic way throughout the world. Now listen here. We are discussing, right, plant breeding, right? Now go for the next topic. Steps involved in plant breeding. Steps involved in plant breeding. Steps involved in plant breeding, right? Now listen here. Plant breeding. Plant breeding. Plant breeding is carried out. Plant breeding is carried out in a systematic manner throughout the world. Whether it is a company or a educational institution, wherever we do this plant breeding procedure or program, we will carry out it in a systematic manner. Therefore, whether, right, in a company or a educational institution, wherever we do, right, plant breeding, it is done in a systematic manner. It is done in a systematic manner. Main steps. Main steps involved in producing a new genetic variety. Producing a new genetic variety by plant breeding. By plant breeding. Or see this statement carefully. Sir, by plant breeding process we are producing a new genetic variety, right? To produce a new genetic variety by plant breeding process, what are the main steps that are involved? Is it okay? Right. Go for number one, first one. Go for the first step. Collection of genetic variability. What is the first step, sir? Collection of genetic variability. Collection of genetic variability. Number two, evaluation and selection of parents. Evaluation and what's our selection of parents. Evaluation and selection of parents. Number three, cross hybridization. What? Cross hybridization. Cross hybridization of selected parents, cross hybridization of selected parents. Number 4, selection and testing, selection and testing of recombinants, selection and testing of what? Recombinants. Next, number 5, test, release, test, release and commercialization. What it is? Test, release and commercialization of new cultivar. What is the 5th step? Test, release and commercialization of new cultivar. These are the main steps that are involved in plant breeding program. Okay, go for the first step. What's our collection of genetic variability? Go for collection of genetic variability. We are discussing the first step. What it is? Collection of genetic variability. Collection of genetic variability. Genetic variability is a root of any breeding program. What it is? Genetic variability. Genetic variability is the root of any breeding program. Any breeding program. Sir, what does this statement mean, sir? See, variation. Variations are due to genes. Yes or no? It means variations are under the control of genes. Variations are under the control of what, sir? Genes. Variations are under the control of what, sir? Genes. Therefore, genetic variability is the root, means nothing but base. Genetic variability is the root of any breeding program, whether it is plant breeding or animal breeding. What is the root of any breeding program, sir? Genetic variability. What it is? Genetic variability. Now listen. In many plants, genetic variability. What it is? Genetic variability. Genetic variability. Genetic variability is available. Genetic variability is available. Where, sir? Genetic variability is available in wild relatives of cultivated plants. Wild relatives of what, sir? Cultivated plants. Now listen this statement carefully. In many plants, genetic variability. What, sir? Genetic variability. Where it is available? In the wild relatives of what, sir? Cultivated plants. In the wild relatives of cultivated plants. Collection and preservation. What and what? Collection and preservation. Collection and preservation of what, sir? Listen. Sir, what is the root of any breeding program? Genetic variability. Yes or no? Right. Sir, but this genetic variability, what? Genetic variability in many plants available in which relatives? Wild relatives of cultivated plants, right? Therefore, collection and preservation of what, sir? Wild plant species. wild plant species. Wild plant species, right? And cultivated relatives of wild species. Cultivated relatives of what, sir? Wild species is a prerequisite. Is a prerequisite for effective exploitation. For effective exploitation of natural genes available in population. Sir, what does this statement mean, sir? Listen this statement carefully. Sir, effectively we want to exploit the natural genes available in a population. To exploit what? Natural genes available in a population effectively. If we want to do this, what we should do? We have to collect and preserve wild plant varieties, species, and cultivated relatives of wild species. Cultivated relatives of wild species. Therefore, collection and preservation of wild plant varieties, species, and cultivated relatives of wild species. If this is done, then only we can effectively exploit the natural genes that are available in a population. What is the meaning of a prerequisite, sir? What is the meaning of prerequisite? If this second part 1, 2 takes place, then this should be done first. If this is done, then only we can do this. What is this? Effective exploitation of natural genes available in a population. When you can effectively exploit the natural genes in a population, sir, when you collect and preserve wild plant varieties, species, and cultivated relatives of wild species. When you do this, then only we can what? Effectively exploit the natural genes available in a population. I think you got this word prerequisite. Is this statement okay? Right? Right. Now listen this statement. Collection. Collection of all alleles. Collection of what, sir? All alleles of all genes. Of all genes in a crop. In a crop is called germplasm collection. Is called germplasm. Germplasm collection. Sir, what is the meaning of germplasm collection, sir? Germplasm collection means we are collecting all alleles of all genes in a crop. Collection of all alleles of all genes in a crop is called as what, sir? Germplasm collection. Germplasm collection. Sir, what is the meaning of alleles, sir? What are these alleles? This concept will be very clear, right, in genetics chapter. Just for your understanding, now I am saying, listen. In living organisms, characters are controlled by genes, yes or no? What are controlled by genes, sir? Characters are controlled by genes. Who are controlling the characters, sir? Characters are controlled by genes. Characters are controlled by genes. Sir, here I am taking one character, sir. One character. Sir, one character is controlled by how many genes, sir? One gene. One character is controlled by how many genes, sir? One gene. Each gene contains two alternative forms. Each gene is having how many alternative forms? Two alternative forms. Therefore, one gene with how many alternative forms? Two alternative forms. Two alternative forms. Sir, what are these two? These two are alternative forms of how many genes, sir? Of one gene. These two alternative forms are called as alleles. What are these two alternative forms of a gene called as alleles? It means one gene contains how many alleles, sir? Two alleles. Therefore, what is the meaning of alleles? Alternative forms of a gene. What is the meaning of alleles, sir? Alternative forms of a gene. Is this concept okay? In living organisms, characters are controlled by genes. In general, each character is controlled by one gene. Each character is controlled by one gene. Each gene with two alternative forms. These two alternative forms of a gene called as what, sir? Alleles. What it is called as alleles. Is this concept okay? Right. Now listen, collection of all alleles of all genes in a crop is called as what, sir? Germplasm collection. Called as what? Germplasm collection. Sir, how we can collect alleles of all genes, sir? See, we are collecting them in the form of either plant or in the form of seeds. Therefore, collection is done, right? Either in the form of what, sir? Either in the form of a plant or in the form of a seed. Therefore, collected. What? Germplasm is collected. What is collected? Germplasm is collected. Germplasm. Germplasm is collected, right? Either as a plant or what, sir? Seed. Either as a plant or as a seed. For example, take a mango seed. If you keep this mango seed in the soil, you will get which plant? Mango plant. It means what are the characters that are present in a mango plant? All that characters are in mango seed. Yes or no? Sir, characters are controlled by genes. Genes are having alleles. It means seed is having characters of mango plant. Mango seed is having characters of mango plant. It means mango seed is having genes and alleles of mango plant. Yes or no? That's why when we are keeping mango seed in the soil, we are getting the same mango plant only, not another plant. Therefore, in the form of seed, we are collecting the germplasm. In the form of plant, we are collecting the germplasm. Is this statement okay? Therefore, germplasm is collected either as a what, sir? Plant or seed. Is this statement okay? Right? Right. This is concerned to the first step. What it is, sir? Collection of genetic variability. What it is? Collection of genetic variability. Now go for second step. What it is? Evaluation and selection of parents. What is the second step? Evaluation and selection of parents. Evaluation and selection of what, sir? Parents. Go for the second step. Now listen here. Sir, we collected the germplasm, right? Now we have to evaluate this germplasm. We have to evaluate the germplasm. We have to evaluate the germplasm. What has to be evaluated? Germplasm has to be evaluated. Why, sir? To identify the plants with desirable characters. To identify plants with desirable characters. To identify plants with desirable characters. Okay, right. We have to evaluate the germplasm. Therefore, germplasm is evaluated. Germplasm is evaluated to identify what's plants with desirable characters. Desirable characters means the characters what we are interested in. Characters what we are interested in. Therefore, to identify a plant, which is having desirable characters, we have to evaluate what's our germplasm. We have to evaluate the germplasm. Is this statement okay? For example, I am taking 4 plants. Taking 4 plants, P1, P2, P3, P4. Plant 1, plant 2, plant 3, plant 4. Okay, right. Sir, plant 1 is having bacterial resistant genes, sir. Having bacterial resistant gene. Plant 2 is having viral resistant genes, sir. Plant 3 is having fungal resistant genes, sir. Fungal resistant gene. Whereas plant 4, it is not having any resistant gene, sir. Not having resistant gene. Not having what, sir? Resistant gene. It is not having any resistant gene. Now here how many plants are there? 4 plants are there. Now listen carefully. Sir, first plant is having which resistant gene? Bacterial resistant gene. Sir, on the plant it will be written, it is bacterial resistant? No. On the plant it is not written. It is bacterial resistant, it's viral resistant, it's fungal resistant. Then how you are saying that this plant is bacterial resistant, sir? By studying the genes, by studying the alleles present in this plant. Yes or no? By studying the alleles, by studying the genes present in this plant, we came to know that this plant is having bacterial resistant gene. It means we are evaluating the genes present in this plant. Similarly, we came to know that this plant is having viral resistant gene. How, sir? By evaluating the genes present in this plant. Yes or no? Right? Therefore, evaluation. What we are evaluating? We are evaluating the genes. We are evaluating the alleles that are already collected in the form of germplasm. Why? To get plants with desirable characters. To get plants with desirable characters. Is this statement okay? Right. For example, see, here how many plants are there, sir? 4 plants are there. You want to produce a plant which is bacterial and viral resistant. For example, you want to produce a plant which is bacterial resistant and viral resistant. Then first what we have to do, sir? First you have to know in which plants that bacterial resistant gene and viral resistant gene are present. Yes or no? So what we have to do? We have to evaluate the germplasm. So go for the first plant. You evaluate the germplasm. It means we are checking the genes present in this plant. Whether we are having bacterial resistant gene or viral resistant gene in this plant or not, we are checking that. We came to know that this plant is with bacterial resistant gene. Now we started evaluating this plant. It means we started checking the genes present in this plant. Sir, this plant, we came to know that this plant is not having viral resistant genes, sir. Be careful. We are intended, we want to produce a plant which is bacterial and viral resistant. It means now we are searching for which genes? Bacterial resistant gene and viral resistant gene. So do this plant contains viral resistant genes, sir? By evaluation of germplasm, we came to know that this plant is not having a viral resistant gene. Now we went for plant 2, PT2. Okay, plant 2. Sir, by evaluation of germplasm, we came to know that this plant is with viral resistant gene. By the evaluation of germplasm, we came to know that this plant is with which gene, sir? Viral resistant gene. Therefore, this plant is with a desirable character. What is that? Viral resistant gene. This plant is with a desirable character. What it is, sir? Bacterial resistant gene. Desirable character. What characters we are interested in? Those are desirable characters. We want to produce a bacterial resistant and viral resistant plant. So we are searching for plants which are having that bacterial resistant gene and viral resistant gene. Is it okay? Right? How we can find out whether plants are having that genes or not, sir? How? By evaluating the germplasm. Therefore, first we have to evaluate the germplasm. By evaluating the germplasm, right, we can find out right plants which are having desirable characters. Now you tell me, in this example, right, what are desirable plants, sir? Plants with bacterial resistant gene, plant with viral resistant gene. Yes or no? Because we want to produce a bacterial and viral resistant plant. So, right, these two are with desirable characters, P1 and P2. These are two plants which are having desirable characters. So we are selecting these two plants as parent plants. We selected these two plants as which plants are? Parent plants. Therefore, see this statement, germplasm is evaluated to identify plants with desirable characters. In this example, desirable characters, bacterial resistant gene, viral resistant gene. Is it okay? How we identified that these two plants are with right bacterial resistant and viral resistant genes, are by evaluating the germplasm. Therefore, after evaluating the germplasm, we came to know that these plants are with desirable characters. Once plants with desirable characters are identified, we are selecting such plants as parent plants. Therefore, plants with desirable characters selected as parent plants. Plants with what, sir? Desirable characters. Selected as a parent plants. Which plants selected as parent plants, sir? Which plants selected as parent plants? Plants which are having desirable characters. Such plants are selected as a parent plants. Is it okay? Sir, now how many plants are there with the bacterial resistant gene? One plant. How many plants are there right having viral resistant gene? One plant. Sir, now we are calling these plants as what? Parent plants. Now the number of plants are less. Yes or no? Only one with bacterial resistant gene, only one with viral resistant gene. So we have to multiply these plants. How to multiply the plants? Therefore, once right we selected plants as parent plants, the parent plants are multiplied. What are multiplied? Parent plants are multiplied. Parent plants are multiplied. Parent plants are multiplied. What are multiplied? Parent plants are multiplied. Is it okay? Right. Pure lines are created wherever possible. If possible, what are created, sir? Pure lines are created. If possible, what are created? Pure lines are created. Therefore, parent plants are made homozygous. Pure lines wherever possible and wherever desirable. Therefore, wherever possible and desirable, their parent plants are made homozygous. Parent plants are made pure lines. What is the meaning of homozygous, sir? What is the meaning of homozygous? I already told you. Each gene with how many alleles? 2 alleles. Each gene with how many alleles, sir? 2 alleles. Now listen this statement. Take a gene by name B. What is the gene name? B. Sir, this B gene now should contain how many alleles? 2 alleles. What are the 2 alleles, sir? One is capital B and second one is small b. Capital B and small b, these are 2 alleles of how many genes? 1 gene. Capital B and small b, these are 2 alleles of how many genes, sir? 1 gene. These are 2 alleles. 2 alleles of 1 gene. 2 alleles of 1 gene. What and what? Capital B and small b. Sir, here I am taking 2 alleles. See. Sir, these are 2 alleles of how many genes, sir? 1 gene. Sir, capital B is a allele. Capital B is a allele. These are 2 alleles of how many genes? 1 gene. Sir, here the 2 alleles of 1 gene same or different, sir? Same. Becomes homozygous. What it becomes? Homozygous. Becomes homozygous. Sir, small b. Here how many alleles are there, sir? 2 alleles. Sir, these are 2 alleles of how many genes, sir? 1 gene. Are they same or different? They are same. Small b. Therefore, this is homozygous or heterozygous, sir? This is homozygous. Now you take capital B, small b. Now see here. Sir, capital B and small b, these are 2 alleles of 1 gene. Sir, here these 2 alleles of 1 gene same or different? Different. What are different? 2 alleles of 1 gene are different. It becomes heterozygous. What it becomes? It becomes heterozygous. Therefore, got the statements. If 2 alleles of 1 gene same, homozygous. If 2 alleles of 1 gene same, homozygous. 2 alleles of 1 gene different, heterozygous. Is this concept clear? Okay, right? Therefore, this is homozygous. This is a heterozygous. Is it okay? Now see here. Sir, here how many alleles are present, sir? 2 alleles are present. But how many types of alleles are present? Only 1 type of allele, that is capital B, homozygous. Sir, number of alleles? 2, sir. Types of alleles? 1, sir. Small b, homozygous. Number of alleles, sir? 2. Types of alleles? 2. Capital B, small b. Is it okay? Therefore, it becomes what, sir? Heterozygous. It becomes what, sir? Heterozygous.